Welcome to all of you. Today we shall discuss something related to the topic reproduction in pteridophytes. A pteridophyte reproduces by three different methods. The first one vegetative, second one asexual, and third one is by sexual method. Vegetative reproduction normally take place by the formation of the bulbils, gemma and fragmentation. These are nothing but the simple primitive methods of propagation which has no biological significance from the genetic diversity point of view. The predominant method of reproduction starting from Rhynia up to the Marsilia is supposed to be the asexual method of reproduction. The pteridophytes reproduce asexually by the formation of spores. Spores are nothing but the single cells that are haploid in nature, and a spore develops into a gametophyte. That means, in a way, a spore is the first cell in the life cycle of a gametophyte. Now, the question arises where these spores are formed. The spores are produced within some specialized sac-like structures called by the name sporangia. Sporangia are nothing but the capsulated bag-like structures that encloses either few or more number of spores. And the spore-bearing organs or the reproductive organs in pteridophytes are called by various names. For example, in Xylotum, it is known as a synangium. In case of Lycopodium salaginella, it is known as strobilus. The spore-bearing complex in case of Equisitum is called by the name sporangiophore or sporophyll, and in majority of the ferns, it is called by the name sori. Of course, in Ophioglossum, it is known as fertile spike, and in case of Marsilia, it is known as so-called sporocarp, and in case of Osmunda, it is known as tassel. Hence, these are nothing but a group or collection of sporangia. Hence, let me take one example. In case of Lycopodium, the Lycopodium sporophyte, when it attains maturity, produces the reproductive organ, that is strobilus, at the tip of the axis. The cone or the strobilus is made up of numerous fertile leaves called by the name the sporophylls, and on the adaxial surface of the sporophylls, usually the sporangia are born, and inside the sporangia, the so-called spores are produced. Coming to the structure of the sporangium, a typical sporangium is made up of a small stalk and an enlarged bag-like or sac-like structure called capsule. And the capsule is covered by means of either single-layered or multi-layered wall. Usually, the innermost layer of the wall is made up of some specialized nutritive rich cells called by the name tapetal cells, and the tapetal cells are supposed to nourish the developing the spores. Now, within the tapetal tissue, the entire region is known as the sporogenous tissue. The sporogenous tissue at a later stage act as spore mother cells, and these spore mother cells, when they attain full growth, undergo meiosis or reduction division, producing spores in tetrad, and these spores are the first cells of the gametophytic generation, and after the full growth of the spores, the so-called sporangial wall breaks open, ruptures, releasing the spores en masse. This is how normally the reproduction takes place asexually in case of Lycopodium. That means reproduction takes place by the formation of the spores. Spores are produced within sporangia. Sporangia are born on the adaxial surface of the sporophylls, and the sporophylls are compactly aggregated to form the so-called strobilus or the cone, which is referred as the reproductive organ. Now, what is the fate of the spores? The spores fall on the ground and they start germination. They undergo division. The first division results in the formation of two cells, an epibasal cell and a hypobasal cell. The consequent divisions result in the formation of a very small multicellular prothallus or the gametophyte, which is usually chordate in outline. It is chordate or heart-shaped in outline. In majority of the ferns, only one type of spores are produced. The condition is said to be homosporous condition. All the spores produced within the sporangia are alike. But in case of Selaginella, sometime in case of Equisetum, in case of Marsilia, two different types of spores are produced. This condition is referred as Hetero Spory. Hence, in Selaginella, we can see the formation of microspores as well as the megaspores, that is the condition known as heterosporous condition. But for this, in majority of the ferns, usually the homosporous condition is seen. And these spores at a later stage develop into a full-fledged gametophyte. The gametophyte may be autotrophic or it may be heterotrophic. The gametophyte may be subterranean or it may be aerial. Gradually, the gametophyte produces the reproductive structures which are called by the name antheridia and archegonia. Antheridia are the male sex organs, whereas the archegonia are the female sex organs. Usually, the antheridia are globose in outline with a very short stalk, and inside this it produces antherozoids, and the antherozoids are also known as the spermatozoids. Whereas the archegonia is almost a flask-shaped structure with a broad basal region called by the name venter and a narrow neck region. And within the venter region, it possesses the most precious egg cell as well as the venter canal cell. At the time of fertilization, the antherozoids are carried along with a thin film of water and they reach the vicinity of the egg. The male and female gametes combine together to form the so-called zygote. The process of fusion of the male and female gametes is called by the name syngamy, and gradually the zygote develop into a sporophyte. Hence, in the life cycle of a pteridophyte, we can see a diplo-haplobiontic alternation of generation. The sporophyte is diploid and the gametophyte is haploid. A continuous heteromorphic alternation of generation is very much witnessed in case of pteridophytes. Thank you.